The mechanism of action of sulfonylureas. The primary mechanism of action of sulfonylureas are to increase insulin secretion from the beta cells in the pancreas. Although there are several other proposed mechanisms of action, this is the one we will be focusing on today. However, before we describe how sulfonylureas work, we must first understand beta cells. This is a diagram of a beta cell of the pancreas. Its primary function is to secrete insulin. There are glucose transporters on the beta cell called GLUT2 transporters that transport glucose into the beta cell. There are also potassium channels and calcium channels that are responsible for regulating ion concentrations and membrane potential. These potassium channels are normally open, allowing potassium to freely exit the cell, and this keeps the cell in a hyperpolarized state. At resting potential, the calcium channels on the cell are normally closed, and calcium cannot enter the cell. And there are also vesicles containing insulin to be released. So when glucose is transported into the cell by the GLUT2 transporters, this glucose is metabolized by the cell into ATP. This ATP binds to the potassium channels, causing them to close. Closure of enough potassium channels causes the cell to become depolarized due to the buildup of positively charged potassium ions inside the cell. Depolarization of the cell causes the voltage-gated calcium channels to open, and this results in an influx of calcium into the beta cell. This influx of calcium triggers the exocytosis of insulin vesicles, resulting in insulin release into the bloodstream and the rest of the body. Now, sulfonylureas, as well as insulin secretagogues such as repaglinide, act by binding to the potassium ion channels, causing them to close. This triggers the depolarization of the cell, ultimately leading to an increase in insulin secretion. So, to summarize what we just went over, an increase in the blood glucose of the body will cause an increase in glucose uptake into beta cells, resulting in more ATP in the cell. This ATP will bind to potassium channels, causing them to close. This triggers depolarization of the cell and opens the calcium channels. An influx of calcium causes insulin release, which results in a blood glucose lowering effect. Sulfonylureas lower blood glucose by acting on the potassium channel section of this flowchart.